students, and welcome back to yet another video tutorial on Drosophila melanogaster embryogenesis. In today's video tutorial, we would be looking at the dorsal and ventral side arrangement in the case of Drosophila melanogaster. Okay, so we know that the process of fertilization in Drosophila takes place after the sperm enters into the egg. So as you know that the male pronucleus and the female pronucleus fuses, the fusion of the male and female pronucleus will lead to the formation of zygote. Now how does the zygote formation takes place, and how does the dorsal and the ventral polarity in the case of Drosophila melanogaster is being produced? हम लोग ये देखेंगे आज किस तरीके से zygote का formation होगा और dorsal और ventral polarity कैसे बनेगी Drosophila melanogaster में. That we would be visualizing.
Transport of the dorsal protein into nuclei is controlled by a cell signaling molecule called spotsel.
Now, the movement of proteins into the nuclei is being controlled by a cytosolic protein called as spazal. Proteins का movement न्यूक्लियस में कैसे होगा, किस तरीके से होगा, इस चीज को कंट्रोल करने के लिए हमारे पास में एक बहुत ही इंपॉर्टेंट प्रोटीन है और उस प्रोटीन का नाम है स्पाजल. वी हैव अ वेरी इंपॉर्टेंट प्रोटीन दैट विल रेगुलेट द मूवमेंट ऑफ proteins into the nucleus of the cells, and that protein is commonly referred to as spazal protein. Now what is this protein spazal going to do? The spazal is going to bind to its receptor, and it is this receptor which will produce a signaling molecule for the transport of certain key important proteins called as dorsal protein. Okay, so dorsal protein is going to move into the ventral nuclei. How? Let us have a look.
Dorsal concentration is highest in the extracellular matrix on the ventral side of the egg cell.
Now, spazal protein concentration is always high towards the ventral side of the egg cell. Okay, when towards the ventral side, the spazal concentration is highest. जब ड्रोसोफिला के एम्ब्रायो के अंदर स्पाजल का कंसंट्रेशन वेंट्रल साइड पे ज़्यादा होता है, तो स्पाजल का कंसंट्रेशन जब वेंट्रल साइड पे ज़्यादा है तो क्या हो जाएगा, वेंट्रल साइड पे ज़्यादा बाइंड करेगी, डॉर्सल साइड पे कम बाइंड करेगी. So the binding of the spazal protein will take place in a much higher concentration in the ventral side. It will take place in a much lower concentration on the dorsal side. So this is the distribution of the protein that is commonly referred to as the spazal protein. As it is going to bind towards the ventral side, जैसे कि स्पाजल प्रोटीन बाइंड कर रही है किस साइड पे, वेंट्रल साइड पे. सो द बाइंडिंग ऑफ दिस स्पाजल प्रोटीन ओवर द वेंट्रल साइड विल इंड्यूस अ सिग्नल ट्रांसडक्शन टू टेक प्लेस. ये क्या करेगा, ये एक सिग्नल ट्रांसडक्शन को करेगा. वंस अ सिग्नल ट्रांसडक्शन इज गोइंग टू टेक प्लेस, then it will cause the delocalization of cytosolic protein into and out of the nucleus. ये कुछ प्रोटीन्स को न्यूक्लियस के अंदर और कुछ प्रोटीन्स को न्यूक्लियस के बाहर करने का काम करेगा. Okay, please pay your attention here. Drosophila is one of the most important topic in developmental biology as well as molecular biology, so please pay your attention here in the class.
This concentration gradient leads to higher concentrations of dorsal protein in ventral nuclei, with lower levels of dorsal protein in nuclei located in lateral regions.
So here you can find that the dorsal protein, high concentration of dorsal protein is going to take place within the ventral nuclei because of the chemical signaling molecules such as spazal. स्पाजल के बाइंड होने की वजह से, स्पाजल के बाइंड होने की वजह से डोर्सल प्रोटीन वेंट्रल न्यूक्लियाई के अंदर चली जाती है. डोर्सल प्रोटीन कहाँ पर चली जाती है, वेंट्रल न्यूक्लियाई के अंदर चली जाती है. So high concentration of dorsal protein began to take place in ventral nuclei. और ventral cytosol में क्या हो जाएगा, क्योंकि ventral nucleus के अंदर चला गया है तो ventral cytosol में क्या हो जाएगा. In the case of ventral cytosol there would be low concentration of the dorsal protein. The dorsal protein will be present at a very low concentration towards the ventral surface. Low concentration of the dorsal protein. Low concentration of the dorsal protein, of the dorsal protein. Okay, so this protein is going to be present in low concentration in the ventral cytosol. Ventral cytosol में ये प्रोटीन काफी कम concentration में present होगी. Okay, so this is how this protein is going to be organized. Then you can find what is going to happen in the dorsal side of the egg. In the case of the dorsal side of the egg, high concentration of dorsal protein is present outside the nucleus. इसमें उल्टा होगा, क्योंकि देखिए there is no spasal protein, there is no spasal. As there is no spasal protein, so there would be no binding to the receptor molecules by any signaling molecules. There no spasal means no signaling, no signaling. As there is no signaling that is going to take place, so what is going to happen? The signal is not going to be perceived by the dorsal nucleus. सिग्नल डॉर्सल न्यूक्लियस तक नहीं पहुंचेगा. जब डॉर्सल न्यूक्लियस तक सिग्नल नहीं पहुंचेगा तो यहां पर जो प्रोटीन थी जिसका नाम था डॉर्सल प्रोटीन, वो कहां जाएगी बताइए? डॉर्सल प्रोटीन कहीं नहीं जा पाएगी क्योंकि कोई सिग्नल आया ही नहीं. यहां पर सिग्नल आ रहा था, तो यहां पर हमने देखा था कि डॉर्सल प्रोटीन यहां पर कहां जा रही थी? बाहर से अंदर की तरफ चली जा रही थी. कहां जा रही थी? न्यूक्लियस के अंदर जा रही थी. और यहां पर क्या हो रहा है? यहां पर बाहर ही रह जाती है. Here the dorsal protein remains outside in the cytosol. So what we find is that high concentration of dorsal protein outside the nuclei, that is in the cytosol, that is in cytosol. Okay, so in the case of cytosol, there would be high concentration of the dorsal protein. Okay, so in the cytosol there would be very high concentration of the dorsal protein that would be present as compared to any other protein. इन प्रोटीन्स का बहुत ही हाई कंसंट्रेशन होगा साइटोसोल में एज कंपेयर टू एनी अदर प्रोटीन. सो व्हाट वी फाउंड हियर इज दैट द डॉर्सल प्रोटीन इज प्रेजेंट इन हाई कंसंट्रेशन ऑन द डॉर्सल साइड, ऑन द डॉर्सल साइटोसोल. डॉर्सल साइड की साइटोसोल में जिस प्रोटीन का कंसंट्रेशन बहुत ज्यादा होता है उसका नाम है डॉर्सल प्रोटीन. ओके, whereas in the case of the ventral nuclei, high concentration of the dorsal protein is present because of the signaling molecule, that is spasal. The dorsal protein is going to move into the ventral nuclei. spasal की वजह से एक सिग्नल कैस्केड ऑफ रिएक्शंस चलती हैं जिसकी वजह से डॉर्सल प्रोटीन वेंट्रल न्यूक्लियाई के अंदर चली जाती है और डॉर्सल सरफेस पे ये नहीं जा पाती न्यूक्लियस के अंदर क्योंकि स्पाजल नहीं है और साइटोसोल में ही रह जाती है. सो इन दिस केस cytosol has high concentration of the dorsal protein. Here the nuclei has the high concentration of the dorsal protein. So this is the basic difference. Here ventral nuclei has very high concentration. Here high concentration is present in the cytosol. Here in the case of cytosol the high concentration is present. This is how the organization of dorsal protein is taking place in the case of dorsal and ventral nuclei. So let us move forward in order to understand the formation of the dorsal and ventral axis and its complete development in Drosophila melanogaster.
This may seem confusing since dorsal protein is found in ventral nuclei.
Okay.
The name of dorsal protein comes from the fact that the concentration of dorsal remains high at the dorsal side of the egg. Since
Now this is very important. The name of the dorsal protein is because the concentration of the dorsal protein remains very high in the dorsal side of the egg. Dorsal side of the egg में concentration सबसे ज़्यादा रहता है इस प्रोटीन का, इसीलिए बेटा इसे क्या बोल दिया जाता है? डॉर्सल प्रोटीन बोल दिया जाता है. जबकि वेंट्रल न्यूक्लियाई में what happens in the case of the ventral nuclei? The dorsal protein enters into the ventral nuclei. As it enters into the ventral nuclei, so on the ventral side of the cytosol, the concentration of the dorsal protein is very low. डॉर्सल प्रोटीन कंसंट्रेशन वेंट्रल साइड पे कम होता है और डॉर्सल साइड पे साइटोसोल में ज्यादा होता है. नेक्स्ट पॉइंट.
none of the dorsal protein has entered the dorsal nuclei. The activation of some genes targeted by the dorsal protein requires peak levels of the dorsal protein, while others can be activated by intermediate and low levels.
Now this is important. It is about the concentration of the dorsal protein. The different types of genes are going to be activated. The nucleus is the same. The genetic material is the same. We know that in the case of Drosophila melanogaster के केस में every nucleus has 2n number of chromosomes, and for that n equals to 4. Okay, that means Drosophila can have 8 number of chromosome at its maximum limit. Okay, so but the genes present over those 8 chromosomes are different. जो जीन्स हैं 8 क्रोमोसोम के ऊपर वो अलग अलग हैं, ठीक है, 4 क्रोमोसोम है और उनका एक पेयर है, तो इसलिए 8 क्रोमोसोम मैं बोल रहा हूँ ड्रोसोफिला में। अब आपको यहाँ पर ये समझने की जरूरत है दैट हाउ द डिफरेंट जीन्स आर एक्सप्रेस्ड इन अवर बॉडी। द डिफरेंट जीन्स आर बीइंग एक्सप्रेस्ड इन अवर बॉडी बाय द डिफरेंट लेवल ऑफ द कंसंट्रेशन ग्रेडियंट रिसीव्ड बाय द सेल्स। हर एक सेल अलग अलग कंसंट्रेशन ग्रेडियंट रिसीव करता है एक प्रोटीन का जिसका नाम था स्पाजल प्रोटीन। स्पाजल प्रोटीन का अलग अलग कंसंट्रेशन जो है वो सारे सेल्स को एक दूसरे से अलग अलग बना देता है। इट इज द कंसंट्रेशन ऑफ द स्पाजल प्रोटीन दैट इज डिफरेंट in the cells from each other, and because of the concentration of this spasal protein, okay, so there is the difference in the expression of genes। स्पाजल प्रोटीन का कंसंट्रेशन अलग अलग होने की वजह से ही यहां पर हम देखते हैं कि जीन्स का एक्सप्रेशन एक दूसरे से अलग अलग हो जाता है। द वन विच रिसीव्स द हाईएस्ट कंसंट्रेशन हैड gotten the activation of the twist genes। देखिए, अगर आप 8 साइड में हैं तो आपका एक्सप्रेशन अलग होगा, आपके बाल होंगे, आप बालों को आगे करेंगे, आप स्पाइक बनाएंगे, ठीक है, मैं भी बना सकता हूँ पीछे से, ठीक है। सो इफ यू आर एन आर्ट्स स्टूडेंट्स यू विल मेक स्पाइक्स, ओके। इफ यू आर अ engineering background and you are going to enter into the IITs, you are going to have a rock and roll, ठीक है, but now you are a biotechnologist, okay, so you are in biotechnology, now there is nothing left, okay, so this will cause the difference in the expression of your genes। See, the choices that we make and the amount of proteins that a cell receives is going to alter the physiology of gene expression। एक सेल को कितनी प्रोटीन मिली, ठीक है, वो उस सेल के एक्सप्रेशन को चेंज कर देगी। आपको भी प्रोटीन मिल जाती, लेकिन 10th के बाद में आपने बायोलॉजी ले ली और उसने 12th में मैथ्स ले ली, इसलिए आपको प्रोटीन नहीं मिल पाई, और बाकी जो अच्छी थी वो आर्ट्स और कॉमर्स में चली गई, इसलिए भी आपको प्रोटीन नहीं मिल पाई। सो योर एक्सप्रेशन चेंजेस। सी, द सेल्स दैट रिसीव्स द हाईएस्ट कंसंट्रेशन ऑफ स्पाजल, उनकी लाइफ में क्या आ जाता है बेटा, ट्विस्ट आ जाता है, ठीक है। जिन सेल्स को सबसे ज्यादा स्पाजल प्रोटीन मिल जाती है उनकी जिंदगी में एक ट्विस्ट आ जाता है, और जिनको मिलती है और थोड़ी मिलती है उनकी लाइफ में आ जाता है रो। सो दिस जीन इज कॉल्ड एज Rho gene, and the third one is referred to as SOG gene, okay। Those who used to use fog perfumes used to have SOG genes। तो आप इसे ऐसे याद कर सकते हो कि सबसे ज्यादा कंसंट्रेशन जहां पर स्पाजल प्रोटीन का है उसमें क्या हो जाता है, then there is a twist on gene। Other pupils are always crying, that is Rho gene, and the third one is the one that is referred to as SOG। SOG represent those genes which do not have spasal protein, okay। इन्होंने परफ्यूम की बोतल खत्म कर दी है, लेकिन इनकी तरफ कोई देखता भी नहीं है। Nobody is going to look towards them, even if they have used the entire perfume bottles or anything like that, but nobody is going to look towards them। Then such type of genes is referred to as the SOG genes। So the three genes are twist on genes, Rho on genes, and SOG on genes। So these three different types of genes will be activated depending upon the level of spasal protein that these three genes gets। Twist genes is only activated when peak level of the dorsal protein is there। Rho gene is activated by intermediate level of the dorsal protein, and SOG gene is activated by the lowest level of the dorsal protein, but still dorsal protein should be there, but repressed by another protein present in the ventral region। Look here,
Other proteins can also act on the genes targeted by the dorsal protein, leading to patterns of expression.
okay।
How well do you understand Drosophila embryogenesis?
Now we will be looking at the questions regarding the Drosophila embryogenesis now। The first lecture is all over, so moving forward,
Leading to patterns of expression. How well do you understand Drosophila embryogenesis? In this section, you will find out.
now please try to answer the questions along with me। Firstly, you must answer the question, then only I am going to explain it to you।
Question 1. How many times do the diploid nuclei divide before cellularization?
So you must know that the diploid number of nuclei are going to divide multiple number of times before the process of cellularization is going to take place. So in how many number of times the nucleus is going to be divided before cellularization, that you will have to answer. Can you look at the book in front of you as well as the lecture that I have provided and answer? So we can say that 13 number of times it is going to.
Correct.
Okay, so you can visualize the solution also. The nuclei undergo 13 cleavage, 10 before they reaches the periphery, and then 3 more divisions are going to take place after migrating to the cortex. Once they have migrated to the cortex, then 3 more divisions are going to take place, and then 13 number of divisions are going to take place once the nuclei have reached towards the cortex layer. Next question. See, this is how the division is going to take place. 8, 9, 10, and then they have migrated towards the periphery. 13 number of divisions have taken place. Moving forward.
Question 2. Which of the following represents an embryo in which the nuclei have been determined to differentiate into specific tissues in the adult fruit fly?
Now you have 4 different diagrams in front of you. You just have to choose one of them. And according to the question, you must identify in which of the following embryo the cells have been irreversibly determined in order to develop into an adult fruit fly. The cellularization of the cells will lead to the irreversible determination of the cells. जब भी सेल्स सेलुलाइजेशन कर जाते हैं, यानी कि सेल्स के अंदर डिवाइड हो जाते हैं, न्यूक्लियस से सेलुलाइजेशन हो जाता है, तभी उनका डिफरेंशिएशन होता है, अदरवाइज नहीं. सो डी इज द बेस्ट ऑप्शन.
Correct.
Okay, so look here. When the nuclei reaches the edge of the cells, they are totipotent, meaning they are capable of forming any other type of cells. Just before cellularization, the nuclei have been irreversibly determined. Before cellularization से पहले nuclei का irreversible determination हो जाता है. They will lead to irreversible determination. Okay, next.
Question 3. What would happen if the Oscar mRNA molecules were deposited at the anterior pole of the egg cell rather than the posterior pole?
Now, the Oscar mRNA, instead of getting accumulated at the posterior pole, has been now accumulated at the anterior pole in the case of Drosophila melanogaster. Drosophila melanogaster के अंदर, instead of accumulating the Oscar in the posterior pole, it has been accumulated in the anterior pole. Now how the differentiation is going to take place, that we must visualize. So what is going to happen here, that we have to see. See, when the Oscar mRNA are present towards the anterior pole, then the germline cells will be produced at the anterior pole. जर्मलाइन सेल्स कहाँ पे प्रोड्यूस जाएंगे, एंटीरियर पोल्स के ऊपर प्रोड्यूस जाएंगे, पोस्टीरियर पे प्रोड्यूस नहीं होंगे. सो दैट वुड बी द बेस्ट आंसर. ओके, सो लेट अस हैव अ लुक. The cells at the posterior end of the cell would eventually differentiate into germ cells. The cells at the anterior end of the egg would eventually differentiate into the germ cells. See this anterior anterior part of the egg is having the Oscar mRNA. So it is this anterior part that will be differentiated into the germline cells. So I will put the answer on B. Cells at both the posterior end and anterior end embryo would develop into an adult fruit fly. No, it would be the best.
Correct.
That is B. Okay, the cells at the anterior end of the cell would eventually differentiate into germ cells. Next point. See this, the division of nucleus is going to take place, and now this is going to form the germ cells. Here germ cells will be present over the anterior part of the body. So look here.
Question 4. You have determined that a particular nucleus on the ventral side of the embryo gives rise to a leg in the adult fruit fly. What would happen if this nucleus was moved to the dorsal side of the embryo before cellularization?
Now see, there is a nucleus which is present, and this nucleus which is present on the ventral side of the Drosophila melanogaster before cellularization has been deleted and has been transferred specifically from the ventral side towards the dorsal side. न्यूक्लियस को उठाया, वेंट्रल साइड से निकाला और डॉर्सल साइड पे उठा के ट्रांसफर कर दिया। एक बार वेंट्रल साइड से ले लिया और डॉर्सल साइड पे ट्रांसफर कर दिया, तो अब क्या क्या चेंजेस आने वाले हैं ये हमें यहाँ पर देखना होगा। व्हाट आर द की स्पेसिफिक चेंजेस दैट आर गोइंग टू टेक प्लेस दैट वी मस्ट विजुअलाइज। What would happen if this nucleus was moved to the dorsal side of the embryo before cellularization? Even before cellularization, if this nucleus was being transferred, then what is going to happen? The nucleus would give rise to a leg, but in the wrong location. So if this was forming the leg, then it will form the leg on the wrong location. The nucleus would become programmed similar to the nucleus in the new location. The nucleus would give rise to a leg in the normal location of the leg. The nucleus would give rise to a leg and the structure produced by the nucleus surrounding it. No, because this is the nucleus. क्योंकि देखिए ये nucleus है, as it is the nucleus, so nucleus is not differentiated. So nucleus will get differentiated. nucleus अभी तक differentiate नहीं हुआ था, अब nucleus का क्या हो जाएगा, differentiation हो जाएगा, और nucleus का differentiation होते ही it will get programmed itself. It has the ability now to get programmed and will lead to the formation of the similar structures that could be produced. लेकिन ये leg नहीं बनाएगा क्योंकि reprogramming हो जाएगी nucleus की.
Correct.
Okay, the nucleus would become programmed similar to the nucleus in new location. How? Look here. Before the process of cellularization, the nucleus was said to be totipotent. अभी तक nucleus अपना क्या था बेटा? Nucleus was said to be totipotent. Why it was totipotent? What is totipotency? The ability of a cell to form the entire cells of the body is referred to as totipotency. किसी भी एक सेल की इस ability को कि वो सारे cells के सारे component को बना दे, उसे totipotency कहते हैं. Meaning they are capable of differentiating into tissues in the adult fruit fly. They have the ability to get differentiated into tissues in the adult fruit fly. When the nucleus is moved, it becomes programmed similar to the nucleus in new location. As the nucleus moves, the nucleus gets reprogrammed. As the nucleus gets reprogrammed, so it will form the same type of structures and no difference, no mutation will be produced in Drosophila melanogaster. Okay, कोई भी mutation produce नहीं होगा Drosophila में moving forward. See this?
Question 5. Which of the following might happen if the cell containing the nucleus from question 4 was moved to the dorsal side of the embryo after cellularization?
Now see, now what the examiner or the question framer wants to ask is, after the cellularization has taken place, पहले before cellularization था, nucleus को ले जा रहे थे, अब की बार cellularization हो चुका है, now cellularization has taken place and now the cell has been transferred. When the cell has been transferred from one side to another, so what is going to happen? So if this cell was forming the leg, then what will happen? The leg formation will now take place over the dorsal surface, okay, and not over the ventral surface. यानी कि डॉर्सल सरफेस जहां पर विंग्स आने चाहिए थे वहां पर क्या आ जाएंगे, लेग्स का डेवलपमेंट हो जाएगा. द न्यूक्लियस विल गिव राइज टू अ लेग बट इन द रॉन्ग लोकेशन द फ्रूट फ्लाई. द न्यूक्लियस विल बिकम प्रोग्राम्ड similar to the nucleus in no location, in new location. No, the nucleus now cannot become programmed because it has been transformed into cell. Nucleus अभी programming नहीं कर सकता क्योंकि ये cell को बना चुका है. The nucleus would give rise to a leg in the normal location for a leg. No, the nucleus would give rise to both a leg and the structure produced by the nucleus surrounding it. No, the nucleus would give rise to a leg but in the wrong location of the fruit fly. Try to
Correct.
answer the questions along with me, okay? See this. So there would be a leg which would be produced over the thoracic part of the Drosophila melanogaster. तो Drosophila melanogaster के thoracic part के ऊपर leg का development हो जाएगा. This is how leg development will take place. Next.
Question 6. The twist gene is only activated by peak levels of the dorsal protein in the nucleus.
Okay.
What would happen?
So there is the dorsal protein. When the dorsal protein is going to move into the nucleus of the ventral nuclei, then it is going to activate a protein called as twist. Secondly, its lower concentration will activate the protein that will be referred to as the next protein called as SOG, and the third protein that would be activated is referred to as twist, SOG and ROCK on. ठीक है. So these are the different types of protein that would be generated. How? The twist gene, what would happen if the concentration of spazole was uniformly high throughout the embryo? If the concentration of spazole is uniformly high throughout the embryo, then what will happen? All cells will express the twist genes. Okay, so this is going to happen. If they are going to be expressed in all the cells, then it will produce the twist genes activated. The twist gene would be activated in all cells.
Correct.
Okay, look here. The spazole, if it is present in the extracellular matrix throughout the embryo, dorsal protein would be transported into the nucleus of the embryo. Therefore, the twist gene will be activated in all the cells of the embryo. Dorsal protein अंदर चली जाएगी और पूरे के पूरे embryo में twist gene का activation हो जाएगा.
Happen if the concentration of spazole was uniformly high around the entire embryo.
See, this is what is going to happen. Okay, if the spazole is uniformly high across the embryo, then the dorsal protein will enter into both dorsal as well as the ventral nuclei. Dorsal protein दोनों में enter कर जाएगा. जब दोनों में enter कर जाएगा तो दोनों में enter कर जाएगा तो क्या कर देगा? Twist genes will get activated in all of them. Okay, just click that option, clearing all the drawings.
Correct.
Okay, moving forward.
After fertilization.
So we are towards the conclusion part of this particular lecture. So just have a view at the conclusion, then you can put out your questions.
The haploid sperm and egg nuclei fuse to form a diploid zygotic nucleus.
Okay, just after fertilization, a diploid zygotic nucleus is produced.
This nucleus undergoes a series of 10 rapid cleavages, during which most of the nuclei migrate to the periphery of the cell. The nuclei then undergo another 3 rounds of cleavage, leading to about 6,000 densely packed, column-shaped nuclei enclosing the central yolk.
And
Cell membranes then form around the nuclei, transforming the egg into a cellular blastoderm.
Then there would be a cellular blastoderm produced. This blastoderm will have a cavity called as blastocele cavity.
When the nuclei reach the edge of the cell, they are totipotent.
Okay.
Just after cellularization, however, the fates of the nuclei have been irreversibly determined to.
After cellularization, after the movement of the nucleus has taken place, and just after cellularization, the fate of each nucleus has been determined on the basis of the presence of the spazole protein. How?
Differentiate into specific tissues in the adult fruit fly. This determination is accomplished through localized mRNA and concentration gradients in the Drosophila egg.
Okay, so it is the localized mRNA molecules such as the Oscar mRNA that will be determining the cell fate. Oscar mRNA सेल के फेट को डिटरमाइन करेगा कि सेल क्या बनाएगा, किसको बनाएगा, कैसे बनाएगा. Concentration gradient of the two protein, one is referred to as the dorsal, another one is referred to as the spazole, also determines cell fate. It is the dorsal protein and the spazole protein that are going to determine the cell fate, that how the cells are going to be dividing. Next.
You have completed this exercise.
With this, we have finished the first exercise of the Drosophila melanogaster. Now, looking at the specific questions, try to ask your questions, your queries, or write it in the chat box.